Welcome back, Focus Learners, to another explainer. Today, we're taking a real deep dive into the world of pharmaceutical manufacturing and quality control. Now, if you're in the industry, you've definitely heard of the ICH Q6A guideline. It's super easy to look at regulatory docs like this as just a set of restrictive hurdles, right? But today, we're going to reframe that completely. We're looking at the general concept section of Q6A, not as a roadblock, but as a strategic toolkit. Think of it as a builder's blueprint, a literal master plan for making sure medicines are safe, effective, and consistently high quality, no matter where in the world they're manufactured.
Okay, let's dive into this by establishing our baseline. What exactly is a specification? Well, according to the guideline, it's a list of tests, references to analytical procedures, and appropriate acceptance criteria. Basically, these criteria are the hard numerical limits, ranges, or other metrics that establish acceptable quality. In everyday terms, conformance to specifications simply means that if you test a drug substance or product using these exact listed procedures, it's going to hit those precise numerical limits. It's the regulatory reality we live in, and honestly, it's the ultimate condition of approval for any new pharma product.
So here is our roadmap for this explainer. We've got 5 stops. First, the Q6A control strategy. Second, smart testing efficiencies. Third, navigating global acceptance criteria. Fourth, managing data and standards. And finally, we'll pull back for the big picture. Don't worry, we're breaking all this dense material down logically. All right, let's kick things off with Section 1, the Q6A control strategy. Now, it's absolutely crucial to understand exactly where specifications fit into the giant puzzle of quality assurance. And this brilliantly illustrates our foundational document, the ICH Q6A Harmonized Tripartite Guideline. Reaching Step 4 of the ICH process, this document is a monumental agreement between regulators in the European Union, Japan, and the United States. It's the global standard for setting a single set of specifications for new drug substances of synthetic chemical origin, and of course the products made from them. It's the definitive rulebook we're unpacking today. But here's a critical nuance the guideline emphasizes right out of the gate. Specifications are just one piece of a total control strategy. You literally cannot test quality into a product at the very end of the line. True quality comes from a much more holistic approach. We're talking thorough product characterization during development, strict adherence to GMP, a fully validated manufacturing process, plus rigorous raw material, in-process, and stability testing. The general concepts make it super clear. Specifications are chosen to confirm quality, not to establish full characterization from scratch.
Moving right along to Section 2, smart testing efficiencies.
So, how do you save resources, time, and money without ever sacrificing patient safety? Well, the Q6A general concepts actually give us some fascinating answers. One major efficiency tool they introduce is periodic or skip testing. Instead of testing every single batch for every single attribute, which can take forever, you test at predetermined intervals. Now, obviously, this isn't a free pass. It requires really solid justification and regulatory approval before you even think about implementing it. It's typically used post-approval for things like residual solvents or microbiological testing in solid oral dosage forms. Basically, once you've conclusively proven your manufacturing process consistently produces high-quality results. Another brilliant efficiency concept is parametric release. Let's juxtapose this with routine release testing. Routine testing relies on end product laboratory testing of every single batch. Parametric release, on the other hand, is a proactive, operational alternative. Take terminally sterilized drug products, for example. Instead of doing a final end product sterility test, you release the batch based on satisfactory monitoring of specific manufacturing parameters, things like temperature, pressure, and time during the sterilization phase. Because these parameters can be measured so accurately in real time, they're actually more reliable in predicting sterility assurance than a final lab test. How cool is that? Then we have the concept of alternative procedures. Sometimes the official pharmacopoeial procedure, the standard harmonized method, might be a really lengthy chromatographic process. But the blueprint allows you to use an alternative procedure for routine release, as long as it controls the quality to an extent that is comparable or superior to the official method. So you might use a much faster spectrophotometric test to release a batch of tablets that don't degrade during manufacturing, while still keeping that official chromatographic procedure on the books to prove compliance during the product's shelf life. This whole efficiency mindset also bleeds into the general concept of in-process tests. The guideline is great because it clarifies exactly when a test done during manufacturing actually crosses over to become part of the formal specification. Think about it like this. If an in-process test is only used to adjust process parameters within an operating range, say checking the hardness of tablet cores before coating, then it's not included in the specification. But if that in-process test is the formal test conducted prior to release and its results dictate whether the batch passes or fails, then it absolutely may be included. It's all about the fundamental purpose of the test. Let's shift gears to Section 3, Navigating Global Acceptance Criteria. Now, what's really interesting about this slide is how the blueprint bridges regulatory divides across completely different regions. When it comes to release versus shelf life acceptance criteria, approaches have historically diverged quite a bit. In the USA, the standard is generally just a single set of criteria applied to both release and shelf life. But over in Japan and the European Union, there's a strict regulatory requirement for distinct, separate specifications for release and for shelf life, assuming those criteria are actually different. The guideline acknowledges these regional quirks while still striving for that overarching global harmonization. And keeping these standards harmonized requires an immense amount of global cooperation. The Pharmacopeal Discussion Group, or PDG, which is made up of the European, Japanese, and United States pharmacopoeias, has a very strict rule. They state, no pharmacopoeia shall revise unilaterally any monograph or chapter after sign-off or after publication. This is huge. It ensures that once a procedure is harmonized, the rug won't just be pulled out from under manufacturers. The references and standards remain truly global.
Okay, let's move on to Section 4, Managing Data and Standards.
So how do we deal with the inevitable constraints, the complexities, and, frankly, the limited data we have when developing a brand new drug? Well, the general concepts speak directly to this reality with Concept 2.5, Limited Data at Filing. When you file a new application, you might only have data from a very small number of batches. The blueprint totally anticipates this. It actually allows for initial specifications to be set based on that limited data, with a clear understanding that they're going to require subsequent review. As you gain more manufacturing experience post-approval, those acceptance criteria might be modified. That could mean loosening them up or tightening them down as the true capability of your process is finally revealed. Another vital standard we have to manage is the reference standard, which is outlined in Concept 2.11. Basically, this is a highly characterized material prepared specifically to be the gold standard benchmark in an assay, identification, or purity test. For new drug substance reference standards used in assays, the impurities have to be strictly identified and controlled, and the purity has to be measured by a quantitative procedure. At the end of the day, your tests are quite literally only as good as the standards you measure them against. To help navigate all these complex rules, the general concepts rely heavily on clear decision trees. Decision tree number 1 is a great example. It guides manufacturers through establishing an acceptance criterion for a specified impurity in a new drug substance. It walks you through this rigorous, logical flow, first determining the impurity level in relevant batches, then calculating the mean plus the upper confidence limit, and asking if the impurity is also a degradation product. It's a perfect representation of the scientific, math-driven logic that underpins these general concepts, making sure safe impurity limits are established completely objectively. And finally, we arrive at Section 5, the big picture. So the crucial point is understanding how all these distinct concepts synergize. The ICH Q6A general concepts aren't just a list of rigid demands or red tape. They form an incredibly dynamic scientific toolkit. They harmonize global standards so manufacturers can build ones and deploy globally. They ensure patient safety and product consistency through a total control strategy. They provide flexible testing alternatives like parametric release and skip testing to really drive efficiency. And maybe most importantly, they evolve with your manufacturing data, allowing specifications to adapt as a product moves from its initial filing into full-blown mature commercial production. Which brings us to a final thought I want to leave you with. The Q6A guideline was a monumental achievement back in 1999. But as we look to the future, as continuous manufacturing and real-time inline analytical technologies become the new standard, how are these established general concepts going to adapt? Will parametric release just become the absolute default? Will skip testing evolve into continuous real-time verification? The blueprint has served us incredibly well, but the next generation of pharmaceutical quality is being drafted right now. Thank you so much for joining us on this explainer, and keep questioning the standards of tomorrow.